Okay, let's jump right into it. Imagine this scenario. You're in the emergency department, and a patient rushes in, struggling to breathe, a severe asthma attack.
Right.
The clinician acts fast, gives them epinephrine, and you see improvement. Great. But then a critical detail emerges. This patient is also taking propranolol, a beta blocker, for mild high blood pressure.
Ah, yeah.
Suddenly, the clinician makes an immediate call. Discontinue the propranolol and switch them to something like verapamil. That single, precise clinical decision right there is our doorway into the incredibly intricate world we're about to deep dive into today.
Indeed. This isn't just, a hypothetical case. It's a perfect illustration of why truly understanding the fundamental language of how drugs interact with our bodies isn't just academic.
Mm.
It's absolutely crucial for making precise therapeutic decisions and, quite literally, saving lives. Our mission in this deep dive is to go beyond the surface, to explore the very molecules that mediate drug effects, and to equip you with the insights that will let you master this information like the elite professionals in the field.
That's right. We're talking about drug receptors and pharmacodynamics, essentially, what a drug does to the body and how it does it. We've pulled together a fantastic stack of sources for this, including key insights from KATZUNG's basic and clinical pharmacology that will take us from the foundational concepts right through to the nuances of clinical application.
Exactly.
So whether you're prepping for a meeting, catching up on a complex field, or just insanely curious about how medicines actually work their magic, get ready for some serious aha moments.
Let's start with a cornerstone,
Yeah.
the receptor. At its essence, what is it? It's the specific component of a cell or organism that directly interacts with a drug and initiates the entire chain of events leading to the drug's observed effects.
Okay.
This fundamental concept, which has been around for over a century, has become the central focus for understanding virtually all drug actions. What's fascinating is how its reach has broadened, now explaining complex biological regulation even in fields like endocrinology, immunology, and molecular biology.
So it's not just some abstract term, it's the master key to how drugs unlock changes in our bodies. And understanding this master key has some seriously important practical consequences, both for how we develop new drugs and for the critical decisions clinicians make every single day, wouldn't you say?
Absolutely. The material we've explored highlights three critical consequences that make receptors so central. First, they dictate the dose-response relationship. The receptor's affinity for a drug, how tightly it binds, determines how much drug is needed to start seeing effects.
Right.
And the total number of receptors available actually limits the maximum effect a drug can possibly produce. It's sort of like a dimmer switch for the drug's power.
Okay.
Second, they ensure drug selectivity. Receptors are the reason drugs choose their targets. A drug's molecular size, its unique shape, and its electrical charge all determine if and how well it will fit into a particular receptor among the vast array of binding sites in a cell. This means that even subtle changes in a drug's chemical structure can dramatically alter its effects, producing both therapeutic benefits and sometimes unwanted toxicity.
Yeah, that makes sense.
And third, receptors mediate the core actions of agonists and antagonists. These are key players we'll break down next.
That's a great setup. So we've hinted at agonists and antagonists. Let's really dig into the fundamental difference between them and why that distinction is so crucial for drug action.
Fundamentally, agonists activate a receptor. Think of them as the on switch. When they bind, they trigger the receptor to signal.
Okay, on switch.
Natural ligands, like the hormones and neurotransmitters your body produces, regulate receptor function as agonists. Some might activate one specific kind of receptor for all their effects, while others can selectively promote just one particular function over another. But you have pharmacologic antagonists. These drugs bind to receptors, but they do not activate generation of a signal. They're the blockers.
Blockers, right.
Their primary role is to interfere with an agonist's ability to activate the receptor. And here's where it gets interesting. Many of the most incredibly useful drugs in clinical medicine are, in fact, pharmacologic antagonists. They work by stopping something from happening rather than starting it.
So one turns it on, the other blocks it. But there's a third fascinating player here. Allosteric modulators. This sounds like they're doing something a bit more nuanced than a simple on-off switch.
Precisely. Allosteric modulators bind to a different site on the receptor than where the natural ligand would normally bind, what we call the orthosteric site.
A different spot. Okay.
By doing so, they don't directly activate or block. Instead, they can subtly change the receptor's activity. Think of them as volume controls or tone knobs. not the main on-off switch.
Ah, interesting analogy.
They can be negative allosteric modulators, reducing receptor activity, or positive allosteric modulators, increasing it. For example, benzodiazepine drugs like diazepam, often used for anxiety, bind to an allosteric site on GABA-activated ion channels.
Right, like Valium.
Exactly. They don't open the channel on their own, but they dramatically potentiate GABA's ability to open it, increasing channel conductance. This modulating effect contributes to their relative safety as they enhance a natural inhibitory signal rather than completely hijacking it. This concept is incredibly powerful because it applies even to orphan receptors, those whose natural ligands are still unknown.
Orphan receptors.
Yeah, take ivacaftor, for instance. It acts as an allosteric modulator on the cystic fibrosis transmembrane regulator, or CFTR ion channel, which is mutated in cystic fibrosis. What's remarkable is that CFTR has no known physiological agonist, yet ivacaftor can still modulate its activity.
Wow.
This opens up entirely new avenues for drug development, targeting receptors in ways we previously couldn't imagine.
Now that we have a handle on what receptors do, let's explore what they are at a molecular level. Are they all the same, or is there a whole molecular zoo out there?
Well, the vast majority of clinically relevant drug receptors, especially those mediating your body's own chemical signals like neurotransmitters and hormones, are proteins.
Proteins, okay.
Historically, we identified and purified these receptors by observing how drugs bound to them. But thanks to massive advances in molecular biology and genome sequencing, we can now identify receptors by their predicted structural homology.
So using the genes.
Exactly. This has unveiled a far greater diversity of receptors for known drugs and, as we just touched on, led to the discovery of those orphan receptors.
So it's not just the classic lock and key proteins we might imagine. What are some of the other surprising things that act as drug receptors?
Beyond those essential regulatory proteins, our sources highlight several other critical classes. First, enzymes. Drugs can either inhibit or activate these molecular machines. Think of methotrexate, which inhibits an enzyme called dihydrofolate reductase, crucial in cancer treatment, or statins, which famously target HMG-CoA reductase to lower cholesterol.
Right, statins are huge.
Then we have transport proteins. These are literally the cellular delivery systems, and they're crucial drug targets. Examples include the Na plus K plus ATPase, which is targeted by cardioactive digitalis glycosides used in heart conditions.
Like digoxin.
That's the one. Or the norepinephrine and serotonin transporter proteins that are the targets of many antidepressant drugs. Even the dopamine transporters, which cocaine acts on, fall into this category.
Okay.
And finally, even basic structural proteins within cells can be drug targets.
Yeah.
A classic example is tubulin, which is the receptor for the anti-inflammatory agent colchicine used to treat gout.
Tubulin.
Hmm.
Interesting. So we know receptors are these crucial proteins and other molecules that interact with drugs to cause effects, but how do we actually measure those effects? How do we quantify the relationship between how much drug you give and the response you get?
That's where pharmacology gets truly quantitative. In carefully controlled in vitro systems, meaning in a lab dish outside of a living organism, the relationship between drug concentration and its effect is often elegantly simple and can be described mathematically. When you plot this, it typically follows a hyperbolic curve.
Hyperbolic, okay.
On this curve, we define two key parameters: Emax, which is the maximal response a drug can produce, the peak effect, and EC50, the concentration of drug that produces 50% of that maximal effect. This hyperbolic relationship actually closely resembles the Mass-Aschen law, which describes how two molecules associate. This led to the fundamental occupancy assumption, the idea that agonists act by binding to and occupying a distinct class of biological molecules with a characteristic affinity. We can also quantify direct receptor binding. Here, Vmax represents the total concentration of receptor sites available.
Bmax, total sites.
And Kd, the equilibrium dissociation constant, is the concentration of free drug at which half maximal binding is observed. The Kd is a crucial measure because it directly characterizes the receptor's affinity for binding the drug. A low Kd means a very high binding affinity.
So low Kd, high affinity. Got it. And EC50 and Kd are related but not always identical, which is an important nuance. And why do we often see these curves plotted on a logarithmic scale, making them look like a stretched out S shape instead of that smooth hyperbolic curve?
That's a great question, and it's purely a matter of practical visualization. The logarithmic transformation simply expands the scale where the effect is changing rapidly at low concentrations, making those critical initial changes easier to see.
Ah, stretches out the beginning.
Exactly. Conversely, it compresses the scale where the effect is changing slowly at high concentrations. It's convenience for understanding the curve's behavior, not a biological or pharmacologic difference in the underlying process.
Okay, that makes sense. Here's where it gets really interesting and a bit counterintuitive. What happens after a drug binds to a receptor? And what's this concept of spare receptors? It sounds almost like the body's being inefficient.
Not inefficient at all, but remarkably adaptable. The process linking drug occupancy to a pharmacologic response is called coupling. When an agonist occupies a receptor, it causes conformational changes, basically. The receptor changes shape, and this is the fundamental basis of activation. For some receptors, like simple ion channels, the relationship between how many are occupied and the response is fairly straightforward. But for others, particularly those linked to complex enzymatic signal transduction pathways, the relationship is much more complex. And that's precisely where spare receptors come in. Receptors are considered spare for a given pharmacologic response if you can actually elicit a maximal biologic response at an agonist concentration that doesn't occupy all the available receptors.
So you're telling me you don't need to fill up every single receptor to get the absolute maximum effect. How do we even know this happens?
It's a fascinating discovery that can be demonstrated experimentally. Scientists use irreversible antagonists to block a proportion of receptors permanently.
Lock some of them off.
Right. Even after blocking a significant number, a high concentration of agonist can still produce a full, undiminished maximal response. It's like having a team of workers. You don't need every single one on the line to hit maximum output if the remaining ones are highly efficient.
Okay.
For instance, your myocardial cells, heart muscle cells, have a large proportion of spare beta-adrenoceptors. Even if 90% are blocked, your heart can still achieve its maximal response to your body's own stress hormones, catecholamines, under the right conditions.
90% blocked and still full response. Wow. So what accounts for this spareness? Is it just that we happen to have too many receptors?
It can be several factors. Sometimes, yes, it's simply that receptors are spare in number relative to the total number of downstream signaling mediators they activate.
Okay.
But other times it's about temporal spareness, the timing. For example, when a beta-adrenoceptor activates a G protein, it can produce a signaling intermediate whose lifetime in the cell vastly outlasts the original agonist-receptor interaction.
So the signal lasts longer than the binding.
Exactly. This means just a few initial binding events can initiate a prolonged and enormously amplified response down the line. It's a brilliant way for the body to amplify signals from very small amounts of ligand.
So what does this all mean clinically? How does this concept of spare receptors impact how we think about drugs?
The concept of spare receptors is incredibly useful clinically because it means that the sensitivity of a cell or tissue to an agonist depends not only on the receptor's affinity, how well the drug binds, but also critically on the degree of spareness. This is a profound lever for precision medicine. You can change the sensitivity of tissues that have spare receptors simply by changing the number of receptors, perhaps through disease or other drug interactions. This allows clinicians to think precisely about drug dosage without needing to understand all the intricate biochemical details. They know there's a built-in buffer and amplification system.
Like a safety margin built in.
Precisely. It makes our bodies incredibly adaptable and offers drug developers a flexible control panel. Mastering this concept is key to elite understanding.
So competitive antagonists bind to receptors but don't activate them. They primarily reduce the effects of agonists. But there are different types of antagonists, right, based on how they behave.
Yes, there are broadly two main classes based on how they interact with agonists. Competitive and non-competitive. With competitive antagonists, if you have a fixed concentration of an agonist and you progressively increase the concentration of a competitive antagonist, you'll see the agonist's response progressively inhibited.
Okay, more antagonists, less effect.
Right. The critical point here is that sufficiently high concentrations of the agonist can actually overcome or surmount the antagonist's effect.
You can outcompete it.
Exactly. What you'll observe is that the maximal effect, the Emax, of the agonist remains the same, but the curve showing the agonist's concentration effect relationship is shifted to the right, meaning you need more agonist to get the same effect. Pharmacologists have precise mathematical tools, like the Shell equation, that allow them to determine how strongly a competitive antagonist binds its key. This lets them predict exactly how much is needed for a given level of blockade.
This has huge real-world implications, especially for drugs we use every day, doesn't it?
Indeed. Our sources highlight two key therapeutic implications for competitive antagonists. First, The level of inhibition depends directly on the antagonist's concentration in the patient's body. Consider propranolol, the beta-adrenoceptor antagonist from our opening scenario. Patients show wide variations in plasma concentrations due to individual differences in how their bodies process and eliminate the drug.
Right. Metabolism varies.
Which means the inhibitory effects on responses to your body's own adrenaline and noradrenaline will vary significantly from person to person. Consequently, doses must often be adjusted individually for each patient.
Makes sense.
Second, the response also depends on the agonist's concentration. The same dose of propranolol might effectively lower a patient's resting heart rate, but have little effect during exercise or stress because the increased release of norepinephrine and epinephrine can overcome the competitive antagonism.
So context matters hugely.
Absolutely. Conversely, a dose that's therapeutic for one patient with hypertension might be toxic for another if their endogenous catecholamine levels are significantly different. It's a dynamic balance.
So what about non-competitive or irreversible antagonists? These sound much more absolute in their action.
They certainly are, in a way. Once a receptor is bound by an irreversible antagonist, the agonist cannot overcome that inhibitory effect, regardless of how much agonist is present. These antagonists often form a covalent or nearly irreversible bond with the receptor.
A permanent bond, essentially.
Or very long-lasting. This typically reduces the maximal effect, the Emax, that the agonist can achieve. However, and this links back to spare receptors, if spare receptors are present, a lower dose of the irreversible antagonist might still allow the agonist to produce a maximal response, but you'd need a much higher agonist concentration to do it.
Ah, connecting the concepts.
Clinically, irreversible antagonists have distinct advantages and disadvantages. Their duration of action is less dependent on how quickly the drug is eliminated from the body and more dependent on how quickly new receptor molecules are synthesized.
Depends on receptor turnover.
Right. For example, phenoxybenzamine, an irreversible alpha-adrenoceptor antagonist used for a rare tumor called pheochromocytoma, is advantageous because it maintains blockade even when the tumor episodically releases massive amounts of catecholamines.
Useful for those bursts.
Exactly. However, if an overdose occurs, it's a real problem, precisely because the blockade cannot be overcome simply by adding more agonist. In such a situation, you'd have to use what we call a physiological antagonist, which acts via a different receptor to counteract the downstream effects.
A different pathway entirely. And just to circle back to allosteric modulators for a second, can they also act non-competitively?
Yes, they can. By binding to a separate site on the receptor, they modify activity without directly blocking the agonist binding site. These interactions are often reversible, and as we discussed, they can be positive or negative modulators. The example of ivacaftor on CFTR is particularly interesting here because it binds to a site entirely separate from any known natural ligand, yet it profoundly impacts the channel's function non-competitively.
We've covered full agonists that switch things fully on and antagonists that block. What about the partial agonists? What makes them different and why are they even useful?
Partial agonists are a fascinating class because even when they occupy all available receptors, they produce a lower maximal response than a full agonist.
A lower ceiling effect.
Exactly. It's like having a light switch that, no matter how hard you flip it, only brings the light up to 60% brightness, even though a different switch could make it 100%. What's key is that their concentration effect curves strikingly resemble those of full agonists that are operating in the presence of an irreversible antagonist that's reduced the Emax.
So their failure to produce a maximal response isn't because they don't bind well or aren't getting to enough receptors.
Correct. It's not due to decreased affinity or poor binding. In fact, partial agonists competitively inhibit the responses produced by full agonists.
So they block the full agonist too.
Yes, this gives them a very interesting mixed agonist-antagonist property. They can partially activate but also block a full agonist if both are present.
Okay, that's complex.
A great clinical example is buprenorphine, a partial agonist of mu-opioid receptors. It's generally considered a safer analgesic than morphine or fentanyl, which are more efficacious or full agonists, because buprenorphine suppresses breathing much less strongly.
Safer ceiling on respiratory depression.
Right. However, this mixed property can also make it anti-algesic when combined with more efficacious opioids, potentially reversing their pain relief. And it can even precipitate severe withdrawal in opioid-dependent individuals, precisely because it competes with and displaces the full opioid, leading to an insufficient level of stimulation for the physiologically adapted nervous system.
We've been focusing on drugs directly interacting with receptors. But are there other ways drugs can antagonize each other or dampen each other's effects without a receptor even being involved?
Yes, absolutely. Our sources highlight two important non-receptor mechanisms. First, chemical antagonism. This is the simplest. One drug simply binds directly to another drug, making it unavailable to interact with its target.
Like magnets sticking together.
Kind of. A classic example is protamine, which is positively charged, directly counteracting heparin, which is negatively charged. They literally bind each other through ionic forces, preventing heparin from interacting with the blood-clotting proteins it normally would. No receptor involved at all.
Simple chemistry.
Second, physiologic antagonism. This involves endogenous regulatory pathways mediated by different receptors. For instance, glucocorticoid hormones increase blood glucose. Insulin, however, physiologically opposes this effect by acting on completely distinct receptor-effector systems to lower blood glucose.
Different systems, opposite effects.
Exactly. While they act on different pathways, clinicians might use insulin to counteract high glucose levels caused by glucocorticoids.
So for clinical use, is physiologic antagonism generally preferred? It sounds like you're just canceling out one effect with another.
Not usually, no. Using a drug as a physiologic antagonist generally produces effects that are less specific and harder to control than using a receptor-specific antagonist.
Less precise.
Right. Let's go back to our heart rate example. To treat a very slow heart rate caused by excess acetylcholine, a specific signal, using isoproterenol, a beta-adrenoceptor agonist that increases heart rate, would be far less rational and potentially more dangerous than simply using atropine, which is a receptor-specific antagonist for acetylcholine at the muscarinic receptor.
Target the source.
Exactly. Atropine directly addresses the cause of the slow heart rate, while isoproterenol would be a blunt instrument, stimulating the heart through a completely different pathway, with potentially unwanted side effects.
Okay, we've covered the receptors themselves and how drugs interact with them. But here's the big question. How do these interactions actually translate into a cell's response? How do we go from a drug binding to a receptor to, say, a change in heart rate or a change in gene expression that lasts for days?
This is where the molecular mechanisms of signal transduction come in the how of it all. The material we've explored outlines 5 fundamental mechanisms of transmembrane signaling.
5 main ways.
Think of them as 5 distinct families of receptor proteins, each having evolved unique strategies to get chemical information from outside the cell across the cell membrane and into the cell's interior. This section helps us answer crucial questions like why some drug effects persist for hours while others diminish rapidly, how amplification happens inside the cell, and where new drug targets can be found. Understanding these is vital for that elite-level grasp.
Let's dive into these 5 fascinating mechanisms, starting with the simplest.
Okay, our first mechanism involves intracellular receptors for lipid-soluble agents. Some biological ligands, like various steroid hormones, corticosteroids, sex steroids, vitamin D, and thyroid hormone, are unique because they are lipid-soluble enough to simply cross the plasma membrane.
Slip right through.
Right. Once inside, they act on intracellular receptors. These receptors typically stimulate gene transcription by binding to specific DNA sequences within the cell's nucleus. The mechanism is quite elegant. In the absence of the hormone, a chaperone protein called heat shock protein 90, or HSP90, binds to the receptor, preventing it from folding into its active conformation.
HSP90 keeps it inactive.
When the hormone binds, it triggers the release of HSP90, allowing the receptor to become active and initiate gene transcription.
So, what are the big clinical implications here for drugs that work this way?
2 major ones. First, because these hormones work by stimulating new protein synthesis, their effects appear after a characteristic lag period of 30 minutes to several hours.
Takes time to make proteins.
Exactly. So, for instance, glucocorticoids won't instantly relieve a severe asthma attack. Second, and this is crucial, their effects can persist for hours or even days after their concentration in the body has dropped to 0.
Effects linger.
Right, primarily due to the slow turnover of the new proteins that were synthesized. Consequently, beneficial or toxic effects usually decrease very slowly when administration of these drugs is stopped. Our second mechanism covers ligand-regulated transmembrane enzymes, including the well-known receptor tyrosine kinases.
RTKs, okay.
This class mediates signaling for hugely important growth factors and hormones like insulin, epidermal growth factor, EGF, and platelet-derived growth factor. PDGF. These are polypeptides, proteins with an extracellular domain for hormone binding and a cytoplasmic enzyme domain. That enzyme domain can be a protein tyrosine kinase, a serine kinase, or even a guanylyl cyclase. The signaling function typically begins when the ligand binds, causing two receptor molecules to join together or dimerize.
They pair up.
This dimerization activates the enzyme activity in their cytoplasmic domains, leading to the phosphorylation of the receptor itself and other downstream signaling proteins. A single activated receptor complex here can powerfully modulate a large number of biochemical processes within the cell.
That immediately sounds like a prime target for cancer drugs, given how central growth factors are to cell proliferation.
It absolutely is. Inhibitors of particular receptor tyrosine kinases are a major class of drugs used in treating neoplastic disorders where excessive growth factor signaling is involved.
Makes sense.
These include large molecule drugs like monoclonal antibodies, such as trastuzumab, which block the ligand from binding, and also small molecule chemicals like gefitinib, which directly inhibit the kinase activity within the cytoplasm. The intensity and duration of action of these agents are often limited by a process called receptor downregulation. This happens when receptors are internalized from the cell surface and then degraded.
The cell removes them.
Right. This process is physiologically essential to limit growth signals, and mutations interfering with it can actually contribute to cancer.
Okay.
However, some receptors, like those for nerve growth factor, are internalized but not degraded. Instead, the signal is transported over long distances inside the cell, which is crucial for neuronal survival and function. Our third mechanism are cytokine receptors. These respond to peptide ligands like growth hormone and interferons, which are key players in immunity and cell growth.
Okay, cytokines.
Their signaling mechanism closely resembles the receptor tyrosine kinases we just discussed, but with one key difference. The protein tyrosine kinase activity is not intrinsic to the receptor itself.
Ah, it's separate.
Instead, a separate kinase from the Janus kinase JG family binds non-covalently to the receptor. After the ligand binds and the receptor dimerizes, these bound jakes become activated. They then phosphorylate specific tyrosine residues on the receptor. These phosphorylated tyrosines then act as docking sites, binding another set of proteins called STATs.
Jakes activate STATs.
Exactly. STATs stands for signal transducers and activators of transcription. The STATs themselves get phosphorylated by the jakes, they then dimerize, and finally they travel to the nucleus to regulate gene transcription. It's a precise relay race for gene control.
Moving on to our fourth mechanism, ion channels. These are absolutely crucial for nerve impulses and muscle contraction, and I know they're huge drug targets. What are the main types involved in drug action that we need to be aware of?
We essentially have two main types that are critical for drug action. First, ligand-gated ion channels. These are directly opened or closed by the binding of a specific ligand, often natural agonists like acetylcholine, serotonin, GABA, and glutamate, all major synaptic transmitters.
Ligand opens the gate.
Right. They transmit signals across the membrane by increasing ion conductance, which rapidly alters the cell's electrical potential. A very well-characterized example is the nicotinic acetylcholine receptor, NACHR, which is a complex protein made of 5 subunits that form a central channel. When acetylcholine binds, it opens this channel, allowing sodium ions to rush in, causing depolarization and triggering a response.
Super fast response.
Exactly. The speed of this signaling, often in milliseconds, is absolutely critical for moment-to-moment information transfer across synapses in your nervous system. These channels can also be regulated by phosphorylation and endocytosis, processes that contribute to synaptic plasticity, fundamental to learning and memory. Our second type are voltage-gated ion channels. These don't bind neurotransmitters directly. Instead, they are controlled by changes in the cell's membrane potential, basically shifts in electrical charge.
Opened by voltage changes.
Right. Drugs that regulate these channels typically bind to a site different from the voltage sensor itself. For instance, verapamil, our example from the intro, binds to a region in the pore of voltage-gated calcium channels in the heart and vascular smooth muscle.
The drug blocks the pore.
Effectively, yes. This produces antiarrhythmic effects and reduces blood pressure without mimicking any natural transmitter. Similarly, local anesthetics like procaine inhibit voltage-sensitive sodium channels, blocking nerve signals. Even channels not strongly sensitive to natural ligands or voltage, like the CFTR we mentioned earlier, are important drug targets. Lumacaftor promotes CFTR delivery, while ivacaftor enhances its conductance. Both are allosteric modulators crucial for cystic fibrosis treatment. And finally, our fifth and arguably the largest and most complex but incredibly common signaling mechanism is through G proteins and second messengers.
The big one.
It is. Many extracellular ligands, from hormones to neurotransmitters, act by increasing intracellular concentrations of what we call second messengers. These are small, rapidly diffusible molecules like cyclic AMP, calcium ion, or phosphoinositides.
CAM-MP, calcium.
Okay? This system has three main components: a cell surface receptor, a GTP binding protein, the G protein, on the cytoplasmic side of the membrane, and an effector element, usually an enzyme or an ion channel, that actually changes the concentration of the second messenger. For the CAM-MP system, for example, the effector is an enzyme called adenyl cyclase, and the G protein involved is Gs.
So the G protein is like the crucial intermediary here, the molecular switch that not only relays the message, but also amplifies the signal dramatically.
Exactly. G proteins rapidly activate their effectors when they bind to a molecule called GTP. But they also have an intrinsic ability to hydrolyze that GTP back to GDP, which then inactivates them.
Built-in off switch.
Precisely. The rate of this hydrolysis determines the duration and amount of downstream responses. This is where the amplification comes in. A neurotransmitter might bind its receptor for only milliseconds, but the activated G's protein can remain active for tens of seconds, enormously amplifying and prolonging that original fleeting signal.
Huge amplification factor.
Massive. There's actually a whole family of G proteins, each mediating effects of specific receptors to distinctive effectors, creating incredible diversity in signaling. And fascinatingly, a single endogenous ligand can bind and stimulate receptors that couple to different subsets of G proteins.
One key, multiple locks.
Sort of. For example, your body's own norepinephrine and epinephrine increase heart rate via GS-coupled beta adrenoceptors, and they constrict blood vessels via GQ-coupled alpha-1 adrenoceptors. This ligand promiscuity, where one ligand can do many things, offers significant opportunities in drug development because we can design drugs that selectively target just one of those effects.
And these receptors are often referred to as GPCRs, the serpentine receptors.
Yes, precisely. GPCRs, or G protein-coupled receptors, are actually the largest receptor family in the human body. They're also known as 7-transmembrane, 7TM receptors, because they're polypeptide chain snakes across the cell membrane 7 times, creating this serpentine-like structure.
Winding through the membrane.
This family includes receptors for adrenergic amines, serotonin, muscarinic acetylcholine, many peptide hormones, and even the receptors responsible for vision. Agonist binding causes a profound conformational change in the GPCR, exposing a cavity that allows the G protein to bind, driving a nucleotide exchange reaction that activates the G protein. Modern high-resolution structural information of these GPCRs is truly revolutionizing drug discovery, giving us unprecedented insight into how they work.
We've explored how these signals are initiated and relayed, but what happens if a signal goes on for too long, or if the body needs to fine-tune its sensitivity? How does the cell actually regulate these powerful responses?
That's where receptor regulation comes in, particularly a process called desensitization. This is where cellular responses attenuate or diminish over time, even if the agonist is still present and binding to the receptor.
The cell gets used to it.
In a manner of speaking, yes. This can be rapidly reversible, allowing for quick resensitization, or it can be more persistent. Multiple mechanisms contribute to GPCR desensitization. A key one involves the phosphorylation of the receptor itself by enzymes called G protein-coupled receptor kinases, GRKs.
Adding a phosphate group.
Right. This phosphorylation essentially puts a brake on the receptor. It diminishes the receptor's ability to activate the G protein, and at the same time increases its affinity for another protein called beta-arrestin.
Beta-arrestin comes in.
Beta-arrestin binding further attenuates the response and can accelerate the removal of the receptor from the plasma membrane, a process known as internalization.
So beta-arrestin is involved in both turning off the signal and getting the receptor off the cell surface. It's like a dual-purpose molecular off-switch and recycling crew.
Precisely. Internalization can lead to downregulation if the receptors are actually degraded in cellular recycling centers called lysosomes. But it can also lead to resensitization if the receptors are dephosphorylated in endosomes and then recycled back to the plasma membrane, ready to respond again.
Rescued and reused.
What's even more intriguing is that beta-arrestin can also act as a molecular scaffold, binding other signaling proteins, adding tremendous flexibility to GPCR signaling. This opens up a new and exciting concept called functional selectivity or agonist bias, where different drugs might selectively promote certain signaling pathways over others from the same receptor. This is a very active area of investigation for discovering new drug actions.
Fascinating. So these G proteins activate effectors that then generate second messengers. We've mentioned them, but let's really dive into the major players here and what each of them does inside the cell.
We have three major, well-established second messenger systems that are critical to understand. First, there's cyclic adenosine monophosphate, or CAMP. CAMP is a ubiquitous intracellular second messenger, meaning it's found almost everywhere inside cells. It mediates a wide range of crucial hormonal responses, from mobilizing stored energy, like carbohydrates in the liver or triglycerides in fat cells, both stimulated by catecholamines, to conserving water by the kidney, regulating calcium balance, and increasing heart rate and contractility.
Some very widespread effects.
Indeed. It also plays roles in adrenal and sex steroid production, smooth muscle relaxation, and many, many other processes. CAMP primarily exerts its effects by stimulating specific enzymes called gamma-dependent protein kinases, often called pKa, yes. These kinases, once activated, transfer a phosphate group from ATP to appropriate substrate proteins, which are often other enzymes. The specificity of CAMP's effects comes from the distinct protein substrates of these kinases that are expressed in different cells. For instance, in liver cells, CAMP-dependent phosphorylation specifically regulates enzymes involved in carbohydrate storage and release.
Specific targets in specific cells. How are these powerful CAMP signals turned off?
The intracellular actions of CAMP are terminated in two main ways: by specific and nonspecific phosphatases that reverse the phosphorylation trigger by the kinases, and critically by enzymes called cyclic nucleotide phosphodiesterase, or PDEs, which degrade CAMP into an inactive form called 5-AMP.
PDEs, break it down.
Right, and this is a huge drug target. Drugs like milrinone, a selective PDE3 inhibitor used for acute heart failure, or even common compounds like caffeine and theophylline, which are competitive PDE inhibitors, all leverage this mechanism to prolong CAMP's effects. Second, we have the phosphoinositides and calcium system. This signaling cascade is triggered by a variety of hormones, neurotransmitters, and growth factors, either through G proteins or receptor tyrosine kinases. The crucial initial step is the stimulation of a membrane enzyme called phospholipase C, PLC.
PLC, okay.
PLC acts like a molecular scissors, cleaving a membrane phospholipid called PIP2 into two distinct second messengers, diacylglycerol, DAG, and inositol 1,7,4-protein-5 trisphosphate, IP3.
DAG and IP3.
DAG stays in the membrane and activates protein kinase C. IP3 is water-soluble, diffuses through the cytoplasm to trigger a rapid and significant calcium, Ca2+, release from internal storage vesicles by binding to specific ligand-gated calcium channels.
IP3 releases stored calcium.
Yes. The elevated cytoplasmic calcium then promotes its binding to calmodulin, a calcium-binding protein, which in turn regulates other enzymes, including calcium-dependent protein kinases. This pathway is considerably more complex than the CAMP pathway, with multiple layers of kinases and specificities, allowing for very fine-tuned cellular responses.
And how is the signal turned off? With all that calcium being released, it sounds like tight control is essential.
It is. IP3 is rapidly inactivated by dephosphorylation, deagiized and metabolized, and calcium itself is actively pumped out of the cytoplasm by specialized calcium pumps. These non-receptor elements are also very important in pharmacotherapy. For instance, lithium, a drug used for bipolar disorder, is known to affect phosphoinositide metabolism, which contributes to its therapeutic actions. Our third major second messenger is cyclic guanosine monophosphate, or CGMP. Unlike CAMP, CGMP has more established and focused signaling roles in specific cell types, such as the retina for vision and intestinal mucosa and vascular smooth muscle.
More specialized roles.
Exactly. Its transduction mechanism closely parallels that of CAMP. Cell surface receptors stimulate an enzyme called membrane-bound guanylate cyclase to produce CGMP, which then stimulates a CGMP-dependent protein kinase.
And what's its most prominent clinical effect, especially in relation to drug action?
In vascular smooth muscle, increased CGMP causes relaxation, which leads to vasodilation.
Relaxes blood vessels.
Right. This happens through a kinase-mediated mechanism that ultimately results in the dephosphorylation of myosin light chains. CGMP synthesis can be elevated by two main mechanisms: atrial natriuretic peptide, ANP, a hormone stimulating a transmembrane receptor, or nitric oxide, NO, a gas.
Nitric oxide, okay.
NO is unique. It's generated in vascular endothelial cells, enters the target cell, and directly activates a cytoplasmic guanylyl cyclase. A number of very useful vasodilating drugs like nitroglycerin and sodium nitroprusside act by generating or mimicking nitric oxide, leading to CGMP production and relaxation of blood vessels. Other drugs, like sildenafil, commonly known as Viagra, used for erectile dysfunction and pulmonary hypertension, produce vasodilation by inhibiting specific phosphodiesterases, specifically interfering with cGMP breakdown, thus prolonging its effects.
So these second messenger pathways interact, but they can also be remarkably isolated or compartmentalized within a cell, leading to very precise effects.
Yes, that's a fantastic point. While the calcium phosphoinositide and KMP pathways can oppose each other, for example, in smooth muscle contraction versus relaxation, or be complementary, such as stimulating glucose release from the liver, their signals can be surprisingly localized.
Local signals despite diffusible messengers.
Right. Despite being diffusible messengers, their effects are constrained due to rapid hydrolysis by phosphodiesterases and the physical scaffolding of signaling components within the cell. This suggests that PDE inhibitor drugs might, in some cases, produce unintended or toxic effects by scrambling these highly localized KMAP signals, disrupting the cell's internal organization. And a common thread that unifies almost all second messenger signaling is reversible phosphorylation. It serves two principal functions: amplification, acting like a molecular memory of activation, allowing a small initial signal to create a large, sustained response.
Amplification.
And flexible regulation, allowing for vastly different effects in different cell types due to differing substrate specificities of the kinases. This is why inhibitors of protein kinases, like trastuzumab and imatinib, show such great potential as therapeutic agents, particularly in cancer, because they can selectively turn off specific amplification steps.
This deep dive has shown us just how complex these molecular interactions are, almost like an incredibly sophisticated inner workings of a clock. How does all this lead to the development of new drugs, and more effective ones at that?
Well, the existence of a specific drug receptor is usually first inferred by carefully studying how a series of structurally similar drugs, what we call congeners, either mimic or antagonize specific effects.
Comparing similar drugs.
Exactly. If a group of agonists consistently shows identical relative potencies for two distinct effects, it strongly suggests that those effects are mediated by similar or even identical receptors. What's truly fascinating is how evolution has created many different receptors to mediate responses to any single chemical signal. For example, the neurotransmitter acetylcholine acts on both fast-acting ligand-gated ion channels, known as nicotinic ACRs, and slower-acting G-protein-coupled receptors, the muscarinic ACRs, sometimes even within the same neurons.
The same signal, different receptor types.
Right. And crucially, each of these structural classes often has multiple subtypes with slightly different properties.
And this incredible diversity, this molecular complexity, is actually a massive opportunity for drug development.
Absolutely. The existence of many distinct receptor classes and subtypes for the same endogenous ligand has opened up hugely important avenues for drug development, allowing for unprecedented selectivity. Think again of propranolol, which is a selective antagonist of beta-adrenoceptors. It can effectively reduce an accelerated heart rate without preventing vasoconstriction, which is mediated by alpha-1 adrenoceptors. This selectivity is what allows us to target specific therapeutic effects while minimizing others. This principle of drug selectivity can even apply to structurally identical receptors that are simply expressed in different cells or tissues.
Same receptor, different place.
Yes, because different cell types express different accessory proteins that interact with the receptors and change their functional effects. For example, tamoxifen, a drug that binds to estrogen receptors, acts as an antagonist in mammary tissue, making it useful for breast cancer. But in bone, it paradoxically acts as an agonist, helping to prevent osteoporosis.
Acts differently depending on the tissue.
Exactly. However, it can also have an unwanted agonist action in the uterus, stimulating endometrial cell proliferation, which can lead to complications. It's a delicate balance.
So new drug development isn't just about targeting the receptor directly anymore. It sounds like we're getting much more sophisticated.
No, not at all. Pharmaceutical chemists are increasingly looking at elements of signaling pathways that are distal to the receptors as targets for selective drugs.
Targeting downstream components.
Right. We've talked about drugs that act on phosphodiesterases and intracellular kinases. There's been remarkable recent progress in developing inhibitors for GTP-binding transducer proteins that operate downstream of tyrosine kinase growth factor receptors. A powerful example is the development of selective inhibitors for a mutated form of KRAS, specifically the G12C mutation found in many tumors. These are now successfully used in lung cancer therapy, targeting a specific downstream molecular fault. And thanks to groundbreaking techniques like X-ray crystallography, we can now literally see how chemically similar agents achieve their incredible selectivity.
Seeing the structures.
Yeah. For instance, beta-1 and beta-2 adrenoceptors have identical orthosteric binding sites, but drugs discriminate between them based on incredibly subtle differences in how they traverse a vestibule or entry passage to access that site. Many GPCRs have such passages, revealing a completely new basis for improving drug selectivity. This ongoing challenge of achieving precise selectivity is also a constant opportunity for developing ever more useful and safer drugs.
We've navigated a truly intricate molecular maze today. Now, to bring it home, how does all this complex molecular detail translate into deciding what drug to give a patient and at what dose in the real world?
This brings us to the crucial link between drug dose and the clinical response you observe in a patient. Clinicians need to understand how these drug-receptor interactions underpin dose-response relationships in actual patients, what causes the variability in how people respond to drugs, and the profound clinical implications of drug selectivity.
Let's talk about those dose-response curves we saw earlier, but now specifically in a patient context.
First, we have graded dose-response relations. These curves relate the dose of a drug to the magnitude of a particular therapeutic effect in an individual. Potency refers to the concentration or dose required to produce 50% of that drug's maximal effect. So drug B might be more potent than drug A if you need less of it to get half the effect, but drug A could still have a larger maximal efficacy.
Potency versus efficacy, important distinction.
Very. Potency depends on the receptor's affinity for the drug and the efficiency of the coupling between binding and response. Clinically, potency is often expressed in practical dosage units, like 50 milligrams for mild sedation. Maximal efficacy is a distinct and often more important parameter. It reflects the limit of the dose response on the response axis, the absolute maximum effect a drug can achieve, regardless of how much you give.
The ceiling effect again?
Exactly. For example, some drugs might have equal maximal efficacy, and all could have greater maximal efficacy than another. This is crucial for clinical decisions, especially when a large response is urgently needed. Maximal efficacy can be determined by how the drug interacts with receptors, as with partial agonists, or by the inherent characteristics of the receptor effector system itself. Remember, in therapeutics, efficacy denotes the extent of an effect achievable in a patient, and can also be limited by a drug's toxicity. You can't give more if it becomes too dangerous. As for the shapes of dose-response curves, while many clinical responses approximate a sigmoid curve when plotted on a logarithmic scale, some do not. Extremely steep curves can have important clinical consequences, especially if the upper portion represents an undesirable or dangerous effect, like coma. Steep curves mean small dose changes have big effects. Precisely. Steep curves can result from cooperative interactions of several different drug actions, making dose adjustments very tricky.
What about when the response isn't graded? You can't measure a little bit more or a little bit less, but rather an either-or event, like preventing a seizure or even death.
For those all-or-nothing outcomes, we use quantile dose effect curves. These curves plot the cumulative frequency distribution of responders versus the log dose in a large number of individuals.
Looking at populations now.
Right. Here we characterize these by the median effective dose, ED50, which is the dose at which 50% of individuals exhibit the specified effect. It's important to note this ED50 means something different from the graded ED50.
Different ED50s, got it.
Similarly, we have the median toxic dose, TD50, and the median lethal dose, LD50. These values are invaluable for comparing drug potencies and understanding their selectivity for different effects across a population. These quantile curves also provide critical information on the margin of safety. The therapeutic index, typically defined as the TD50 divided by the ED50 in animal studies, estimates the drug's potential benefit versus its risk.
Therapeutic index, toxic dose over effective dose.
More practically in humans, we think about the therapeutic window, the range between the minimum toxic dose and the minimum therapeutic dose. The acceptable risk of toxicity critically depends on the severity of the disease being treated.
Risk versus benefit trade-off.
Exactly. For instance, the dose range that provides relief from an ordinary headache should have a much wider margin to serious toxicity than the acceptable difference for treating a lethal disease like Hodgkin lymphoma, where more toxicity might be tolerated for a life-saving outcome. Both graded and quantile curves are incredibly valuable. Grading curves tell us about a drug's maximal efficacy in an individual, while quantile curves reveal the potential variability of responsiveness among different individuals in a population.
This is where the complexity of the human body really comes into play. Why do people respond so differently to the same drug, sometimes in surprising ways?
Individual variability is considerable, and even a single person can respond differently over time. Sometimes it's an idiosyncratic response, an unusual, unexpected reaction, often with a genetic or immunologic basis.
Unpredictable reaction.
More commonly, it's quantitative. A patient might be hyperreactive, needing more of the drug for an effect, or hyperreactive, responding strongly to a small dose. Then there's tolerance, where responsiveness decreases with continued administration, and tachyphylaxis, which is a rapid, acute diminution of responsiveness after just a few doses.
Tolerance versus tachyphylaxis.
Right. Our sources identify 4 general mechanisms contributing to this widespread variation. First, alteration in the concentration of drug that reaches the receptor. This is about pharmacokinetics. Differences in how patients absorb, distribute, metabolize, or clear a drug can profoundly alter the concentration of the drug that actually reaches the receptors.
How the body handles the drug.
Exactly. These differences can often be predicted by factors like age, weight, sex, disease state, especially liver or kidney function, and increasingly by pharmacogenetics, the study of how genetic differences in drug-metabolizing enzymes influence drug response. Even active transport mechanisms mediated by multi-drug resistance, MDR genes, can influence drug availability, often contributing to tumor resistance to anti-cancer drugs. Second, variation in the concentration of an endogenous receptor ligand. This heavily impacts responses to pharmacologic antagonists.
Back to the agonist concentration.
Right. Our propranolol example illustrates this perfectly. It dramatically slows heart rate in a patient with highly elevated endogenous catecholamines, like someone with pheochromocytoma.
Right.
But it has little effect on a marathon runner's already low resting heart rate because there's less endogenous ligand to block.
Depends on the baseline signal.
Precisely. Partial agonists can show even more dramatic differences. For example, psorallicin lowers blood pressure in hypertensive patients with high angiotensin II levels, but it can paradoxically raise blood pressure in patients with normal levels because it acts as a weak agonist where there isn't a strong endogenous signal. Third, alterations in the number or function of receptors. Changes in the number of receptors, either increasing or decreasing, or changes in their coupling efficiency to downstream pathways can significantly alter drug response.
Changes in the target itself.
Yes. For example, thyroid hormones increase the number of beta-adrenoceptors in the heart, making it more sensitive to catecholamines, which explains why propranolol is useful in thyrotoxicosis. Agonists themselves can induce downregulation or desensitization of their receptors, directly contributing to tachyphylaxis or tolerance. This also explains overshoot phenomena upon drug withdrawal. Discontinuing an antagonist might lead to an increase in receptor numbers, causing an exaggerated response when the drug is removed.
Rebound effects.
Exactly. Conversely, discontinuing an agonist might leave too few receptors, leading to insufficient stimulation, like the dangerous hypertensive crisis that can occur with clonidine withdrawal. This area is a prime focus for personalized or precision medicine. Genetic factors identified through pharmacogenetics can increasingly predict drug response. For instance, specific mutations in the epidermal growth factor receptor in certain lung cancers can dramatically enhance sensitivity to kinase inhibitors like gefinib, significantly improving the therapeutic index for these patients. And fourth, in a very broad category, are changes in components of response distal to the receptor. Even if a drug binds perfectly to its receptor, the final physiological response depends on the integrity of all the biochemical processes within the responding cell and the overall physiological regulation by interacting organ systems.
Everything downstream matters too.
Absolutely. This is arguably the largest and most important class of mechanisms causing variability. The most crucial cause of treatment failure is often a wrong or incomplete diagnosis. Drug therapy works best when it's precisely targeted at the underlying pathophysiology. Even with a correct diagnosis, compensatory mechanisms in the patient like increased sympathetic tone or fluid retention opposing antihypertensive drug can diminish beneficial effects, often requiring additional drugs to achieve the desired outcome.
So given all this incredible complexity, it sounds like no drug just does one thing, right? How do we, as clinicians and as informed individuals, manage the good effects versus the potentially bad ones?
You've hit on a truly critical point. No drug causes only a single, perfectly specific effect. Drugs are inherently selective, not truly specific.
Selective, not specific. Key difference.
It is. They bind to one or a few receptor types more tightly than others, and these receptors control discrete processes that result in distinct effects throughout the body. It's precisely because of this selectivity, not absolute specificity, that drugs are useful. We typically categorize effects into beneficial or therapeutic versus toxic or adverse.
And toxicity can arise in different ways, even from the good receptors, the ones we're trying to target.
Yes, our sources outline 3 main scenarios where this happens. First, beneficial and toxic effects mediated by the same receptor-effector mechanism. Much serious toxicity is a direct, dose-dependent extension of the drug's therapeutic actions. Think of bleeding from anticoagulants or hypoglycemic coma from insulin. Both are simply too much of the desired effect.
Too much of a good.
Exactly. These can often be avoided by careful dose management, rigorous monitoring, or if the indication for the drug is weak, simply not administering it. If the drug is necessary but causes unacceptable toxicity, adding another drug acting through a different mechanism often allows for reducing the dose and thus the toxicity of the first drug. Second, beneficial and toxic effects mediated by identical receptors but in different tissues or by different effector pathways. Many drugs act on a single receptor type but produce both desired and undesired effects simply because that receptor is present in different tissues.
Right place, wrong place effects.
Examples include digitalis glycosides, which target NA plus K plus ATPase in both the heart and elsewhere, or methotrexate, which targets dihydrofolate reductase in cancer cells, but also healthy cells. Glucocorticoid hormones are another classic example. Strategies to mitigate this toxicity include administering the lowest effective dose, using adjunctive drugs with different mechanisms and toxicities, for instance, combining other immunosuppressants with glucocorticoids. Or, critically, manipulating drug concentrations available to different tissues, like using aerosol glucocorticoids for asthma, which localizes the drug to the bronchi and minimizes systemic effects.
The local delivery helps.
Third, beneficial and toxic effects, mediated by different types of receptors. This is where improved receptor selectivity truly provides a therapeutic advantage. Many receptors and their therapeutic uses were discovered by analyzing the effects of physiological chemical signals, and this continues to be a fruitful area for discovery. Other drugs were discovered by simply observing the effects of chemically similar agents in clinical contexts, like opioids or antipsychotics, and only later were found to interact with specific endogenous receptors.
Finding the target after finding the drug.
Sometimes, yes. Modern structural biology is deepening this understanding significantly. For example, X-ray crystallography shows that your beta-1 and beta-2 adrenoceptors have identical orthosteric binding sites, but drugs can discriminate between them based on incredibly subtle differences in how they traverse a vestibule or entry passage to access that site. Many GPCRs have such passages, revealing a new basis for improving the selectivity of GPCR-targeted drugs.
So the constant challenge of managing unintended drug effects is also the driving force for pharmaceutical innovation, pushing us to understand these molecular details even better.
Couldn't have said it better myself.
Wow, what a deep dive. We've journeyed from the very definition of a drug receptor through the intricate molecular dance of agonists, antagonists, and all the sophisticated signaling pathways within our cells. We then saw how these micro-interactions manifest in real-world patient responses and how this entire understanding fuels the endless quest for safer, more effective drugs.
Truly understanding receptor affinity, maximal efficacy, the profound phenomenon of spare receptors, the diverse signaling mechanisms, and the myriad factors that cause variability in patients. These are the true hallmarks of mastering pharmacology like an elite professional.
It all ties together.
It does. It profoundly eliminates why the initial clinical decision to immediately discontinue propranolol in our asthma patient was so critical. Propranolol's beta-adrenoceptor antagonism prevents bronchodilation, potentially exacerbating the asthma. In contrast, verapamil, a calcium channel blocker, acts on a completely different target, avoiding that specific respiratory risk. Or why, if the patient's asthma was a known factor, a more selective beta-1 antagonist like metoprolol would have been a superior choice from the start.
Understanding the mechanism avoids the harm. Here's where it really makes you think. If we consider how drugs leverage these incredibly complex and often redundant signaling pathways within our bodies, and given the rapid advances in AI and systems biology, what kind of radical new drug development might we see emerge in the coming years? Will we move beyond simple single receptor targeting to optimize multi-pathway modulation for truly personalized medicine?
That raises a truly important question, doesn't it? How might our ever-deepening understanding of these nuanced interactions allow us to design drugs that don't just block or activate, but finely tune cellular responses, taking into account an individual's unique biological landscape in ways we've only dreamed of?
That's something profound for you to mull on your own. We hope this deep dive helps you feel more informed, more curious, and certainly better equipped to navigate the fascinating and ever-evolving world of pharmacodynamics.